Today, we will be showing you just how extraordinary octopuses are. Welcome to Extraordinary Octopuses, brought to you by Lauren, Michaela, Nasia, and Orshi. But what makes octopuses so interesting? Well, to start, they have similar nervous system development genes as those found in intelligent vertebrates, like some humans. One highly copied group of genes contain instructions for making protocadherin proteins. Having a greater variety of these proteins, which play a role in nervous system development, enables the construction of more complex neural networks. They also have the ability to navigate mazes, solve complex problems, remember, predict, and use tools to take apart just about anything. They have not one, not two, but 3 hearts. What we are going to focus on today, though, is their unique ability to edit their RNA.
We are going to be specifically focusing on this article, Octopuses and Squid are masters of RNA editing while leaving DNA intact. Cephalopods, like this little guy here, excel at a type of editing known as adenosine to inosine, or A to I editing. This happens when an enzyme called ADAR2 strips a nitrogen and 2 hydrogen atoms off adenosine, the A. That chemical peel turns adenosine into inosine, the I. Ribosomes read inosine as guanine instead of adenine. Sometimes that switch has no effect on the resulting protein's chain of amino acids, but in some cases, having a G where an A should be results in a different amino acid being inserted into the protein. Such protein-altering RNA editing is called RNA recoding.
RNA editing isn't just limited to creatures of the deep. Almost every multicellular organism has one or more RNA editing enzymes called ADAR enzymes, short for adenosine deaminase, that acts on RNA. But those edits rarely change the amino acids in a protein. For instance, Ellie Eisenberg of Tel Aviv University and her colleagues identified more than 4.6 million editing sites in human RNAs. Of those, only 1,517 recode proteins, the researchers reported last year in Nature Communications. Of those recording sites, up to 835 are shared with other mammals, suggesting that evolutionary forces preserved editing at those locations. In a common form of RNA editing, an adenosine becomes an iosine through a reaction that removes an amino group and replaces it with an oxygen. The picture on the right shows an ADAR enzyme attaching to a double-stranded RNA at the dsRNA binding domain. The region of the enzyme that will interact to cause the reaction, the deaminase domain, is positioned near the adenosine that will become an iosine.
Cephalopods take RNA recoding to a whole new level. Longfin squid, for example, have 57,108 recoding sites, which was reported in 2015. Since then, researchers have examined multiple species of octopus, squid, and cuttlefish, each time finding tens of thousands of recoding sites. Soft-bodied or coleoid cephalopods may have more opportunities for editing than any other animals, because of where at least one of their ADR enzymes is located in the cell. The specific enzyme is the ADR2 enzyme. Thank you for your attention, and make sure that you give octopuses the respect they deserve.